afternoon, everyone. Welcome back to the next session. This is the Bootcamp number 2, Discovery and Preclinical Development Challenges. My name is Mark Albrecht. I'm the chief of the antibacterials branch at BARDA, and it's really exciting to be here with you this afternoon to talk to you and hear these presentations today that are really going to focus in on the challenges that we face during early development of products. Now, of course, BARDA looks at the late-stage development challenges. Here we're going to focus in on those early-stage challenges, technical challenges that face a developer as they move into this space. We've got 3 excellent presenters this afternoon, and I'm really looking forward to hearing from each one of them. The first one is Heike Brötz-Oesterhelt. Hopefully I pronounced that close. There we go. Excellent. We're going to be able to hear from Dr. Brötz-Oesterhelt, who hails from the University of Tuebingen in Germany. Really looking forward to hearing her presentation, focusing in on characterizing a good target. With that, turn it over to you. Thank you.
Yeah. I just would like to make you aware that this session will be recorded. Thank you.
Yes, thank you very much for this nice introduction. Thank you for inviting me, and this will be a shift of gears. It will no longer be about funding opportunities or big organization. I think this will be much more down-to-earth fundamental science, and I apologize also because I recognize many known faces, dear faces in the audience, because I thought it might be something here today as the GRS before the GRC, and I made it a bit educational. So in case you know all this, what I'm going to tell you, I think it might be at least be a nice basis for some discussion and some, summary about the topic. So yes, what I will be talking about is what is a good target and how can we start studying good target. My background, I have been working in antibacterial discovery and particularly modes of action for more than 25 years. And I'm always as long in academia now as I have been in industry before, so I have been both sides. But my personal love has always been mechanistic studies on the targets, or basically I rather prefer to say not the targets, because that always reminds you only of a single enzyme, but I would say modes of action, because they are often more complex than a single target. And over the years we have established something that I call a mode of action discovery platform with a variety of assays that are already available and handy for studying mechanistic details of antibacterial activity. In my department that I had, we have also a colleague working on the biosynthesis of natural products, which is a very nice complementation. We have a colleague working on the physiology of streptomycetes, which have been, over the last decades, the prime family of organisms producing antibiotics, and we have a natural product chemist, and therefore my talk will also be a pledge towards natural product antibacterials. So what is a good target? If you had asked this question in the year 2000, probably a colleague at a company, and at that time I was at Bayer, would have given you this list of characteristics that you can read here: new, broadly conserved, essential, selective, screenable, druggable. And I say yes, all of this is a good target, but a good target is so much more than that. And that is what my talk will be about, because it turned out then just getting a new target, a new enzyme that has all these characteristics on this list, and find a binder to this target or an inhibitor of its enzymatic function even, is not enough to make a good drug. And yes, you know all these very great reviews that have been published. There have been lots of screens, and so these target-based assays didn't bring that many new starting points for development programs as we would have wished at that time. One thing that is related to this is not related to the targets as such. So we will be talking most about the target in my presentation, or targets, but please allow me just this one excursion towards the compounds, because it's always the compound eventually that makes the drug. And what occurred during the screens at that time is that the compound has to enter the cell in order to inhibit the target. And that means that you have also to consider the screening formats, and that, yes, the compound really needs to be available and engage the target inside of the bacterial cell, and you can't just use standard libraries of compounds that had been, the synthetic libraries that had been available in the companies at that time, and that had been screened over and over again. And this grayish, grayish cloud that you see here, you see that on the upper left corner here in this diagram. This has been a great study by Archaeogen, where they compared all the drugs that are used against various indications, spanning cardiovascular, anti-infectives, but also CNS and whatsoever. And these are all residing in this diagram, where the hydrophobicity, the log D value as a measure of hydrophobicity, is plotted against the molecular weight, and they cluster in this upper left corner. While you see that the antibiotics, the blue ones are the ones targeting gram-negatives, and the red ones are the ones targeting gram-positive. They cover a different chemical space, and that is also what got the idea towards COAD, yes, and the academic discovery and screening activity of a broader structural range. So why is that it seems to be difficult to enter bacterial cell, and that is more particular a problem for gram-negative bacteria? And the reason is, and others have studied this much more in depth than me, that we have two opposing barriers. The one is the outer membrane, and for a molecule to enter or to pass passively the outer membrane, it needs to be hydrophilic and preferentially cationic and have a small molecular weight up to, for E. coli, 600 in order to be able to pass the porin. But when it has reached the periplasm, what shall it do with these characteristics? In order to passively pass the cytoplasmic membrane, it needs to be hydrophobic. And this is quite the opposite. So you will learn all the antibiotics that we have, they have very specific ways of making their way into the bacterial cell. And this is often a story of each molecule, and every molecule does it in a different way. And currently there are being studies, the rule of uptake, and Paul Hergenrother is doing pioneering work in that regard, and Lynn Silver has always been describing this. One recent project that we had is we were studying the antibiotic negamycin. It is a natural product. It is a peptide or a pseudopeptide that has been around for some time, where a range of companies have tried to optimize but often fail to improve the activity. It's binder at the ribosome, binding due close to the 8 site with a novel mechanism and without cross-resistance, a very interesting molecule. And we have been looking more in depth into the uptake of the molecule into the bacterial cell to better understand, because we think particularly for a molecule like this, uptake is as important as optimization at the target. And we found actually that this molecule has a number of uptake routes. And so for once, because it's pseudopeptide, it is nicely recognized by ATP-dependent peptide transporters, but not only such. And there is more than one. There are also so-called POTS, proton-dependent oligopeptide transporters, that it can use. There are transporter-independent uptake modes, and we found that there can be passive diffusion in a certain constellation at a certain pH range when it can be a Zwitter ion. And calcium helps the molecule to interact with the phospholipids. And interestingly, so there is very low resistance rates. So the resistance rate is smaller than 10 to the minus 9, while for other peptides that can only use one tran uptake route, there is rapid resistance development. So here you can reach up like 2-fold MIC or 4 times MIC, but not higher. And that just shows, in the talk about target, I want to put that beforehand to say, like, yes, the compound has to reach the target, and uptake is an important point. So now coming to the targets. If you want to screen for new molecules, you better work with whole cell assays, because you have already the uptake issue included and implemented. And focused approaches always beat random screening. So what are the targets of successful antibiotics that we apply in the clinic? And you see them all here. This is a scheme from a textbook. But if you look at them more closely, you will see they are not only enzymes. So not in a sense that you have a soluble isolated enzyme taking its metabolite and making a conversion of the metabolite, and the compound is just inhibiting this isolated enzyme. No, many of those are part of big machineries. We have the ribosome, or the area to the right. We have cell wall synthesis, which is a very large machinery. DNA gyrase running at the front of the replication fork. The RNA polymerase is a big machine. Or we have molecules binding to phospholipids or peptidoglycan precursors in their form as lipids. So it's not just that you consider there is an enzyme and only I have to inhibit this enzyme. And why do you think? Because many of these antibiotics have been derived from natural product. Why do you think nature has picked particularly such kinds of targets? One thing is, the complexer the machinery, the more trouble you can make by disturbing it. And so it's not so much that you most strongly inhibit the particular enzyme reaction. Maybe you insert in a certain place, or you disturb maybe the membrane, or you prevent the interaction, and all this will help to make a powerful mechanism. So powerful mechanisms can disturb spatial order. They can affect and disturb lipid environments or protein environments. And like 20 years ago, it was a no-no to try to go with a small molecule to inhibit a protein-protein interaction, but that has been shown that this works too. Also, another machine or a complex system is the ribosome, and that is like a hotspot for antibiotic action. If you remember this big a long list, yes, to the right on my previous slide. So why has nature selected the ribosome as such a good target? One thing is binding to almost all compounds, yes, all compounds, they bind to ribosomal RNA. So why is that an advantage as opposed to bind to a ribosomal protein? The advantage is that E. coli has 7 copies of the DNA encoding the ribosomal RNA. So you will not get rapid resistance development by mutation of the target gene. So when you want to develop resistance, you have to get an RNA methylase, yes, to methylating all the RNAs of the ribosome, but not a point mutation in the target. That may be one advantage. If you compare also two 30S binders, tetracyclines and aminoglycosides, and they both bind close to the A sites. Tetracyclines cause translation arrest, while aminoglycosides, they cause mistranslation. The first gives you a bacteriostatic response, the second a bactericidal. And why is that? Because the cell obviously can much better cope with the situation where translation is just stalled because it knows it from nature. Sometimes there is in the environment an amino acid lacking from the nutrients, and then the cell goes into a stringent response. While when you make rubbish proteins, there is so much, such a mess in the cell, and that affects every pathway, and that is a pleiotropic destructive event that follows, is downstream of the actual target interaction. And I think this is something that you should consider when you discuss potent mechanisms of antibiotics. Go beyond that thinking of a target as an isolated enzyme in the test tube. Go beyond that thinking of just measuring inhibition constants, but think about all the downstream process and cascades that are affected. Lessons learned from therapeutically validated targets. Yes, as I said, targets of successful antibiotics that are used for a long time in broad spectrum systemic therapy are not encoded by single genes. And for instance, beta-lactams, more than one penicillin binding protein, quinolones, 2 topoisomerases, glycopeptides, it's not a protein at all, it's a peptidoglycan precursor. Ribosomal inhibitors, as I said, binding to the RNA within the ribosome. There are, of course, compounds that we also use that bind to single gene targets, but you better protect them in combination therapy. We have also learned with fundamental research that small molecules can have polypharmacology. They can have more than one target. And yes, complex mechanisms, as I explained to you on the example of the aminoglycosides, are better in causing disruptive, better achieving bactericidal mechanisms. Right. So coming from that, most of the molecules or classes from the current list on the previous slide have been natural products. So I think there is no doubt up to that point, until we do better and understand better with rational design, that natural products make good starting points for antibacterial discovery. And the reason is embedded in their different structures, their complex structures. They are more stereocenters, conveying the opportunity for polypharmacology, hydrophilicity, and yes, I think this is something most of you probably have heard about. The problem with current natural product research is that we are currently at a time when unless we look at very new and different sources, we rediscover molecules that we have already known. And that takes lots of effort. And so what can we do in order to help here? I mean, there had been tremendous progress in mass spec technology, high resolution MS, and there are databases where the chemists, when they look into an extract on the basis of the mass that they get, which is 4-digit behind the comma, can already give a guess what this molecule might be in that it causes the antibacterial activity, if that is a known one. Here we have been working in order to complement this by a biological approach, bioreporters that have been selected in a long ago screening program on the basis of a full genome transcriptome study of Bacillus subtilis with 100 different antibiotics, and the colleagues at Bayer at that time, they picked promoters that are selectively induced if a certain mechanism of action acts on these cells. Like for instance, if you say I have a translation staller, then I get a certain reporter induced, as opposed to I have a transcription inhibitor, or a problem that gives the cells SOS response and DNA strand breaks. So we have been using these reporters. We put beta-galactosidase behind a whole suite of these bioreporters, and we validated them with 100 reference antibiotics. And when that was done, we started on a pilot project with the Tuebingen strain collection of producer strains, and we wanted to see how these bioreporters fare and if they give any additional information on top of this screening for the masses. And what we first found is, like the natural product researchers worldwide, what they often do is they apply an extract, or they take the producer strain and they look for inhibition zones in the agar against the bacterium of interest that they want to inhibit. And with with our bioreporters, they can do exactly the same, but they do not get only an inhibition zone, they get a blue halo that tells them if the inhibitor that they found may maybe store the ribosome. And if you have a certain suite of such bioreporters, then you get already mode of action information that is intrinsic to the very same assay that they normally do. And if now the colleague at the MS sees this particular mass, where that he knows is an anthraquinone, and he said this might bind to the DNA, and my DNA stress reporter sets out, then that is already a confirmation. So these bioreporters work well along the entire purification pipeline in natural product discovery, from the producer strains directly that you saw on the previous slide without any purification, along the extracts or the fractions. You can see in which fraction the bioreporter activity is that you would want to see, up to the pure compound. And for instance, here is an interesting opportunity. What natural product researchers often do is they screen first on solid medium and get the inhibition zone, and then they grow the producer strain in liquid medium because it's much easier to purify it from liquid medium. And then they say, oh, I still see the same very nice halo, and then I now go for the production in liquid, right? Because it's easier for me than scratch all the agar plates and extract from agar. But here you see, only in the agar you would have gotten the bioreporter activity. The producer is making something else in liquid. And the reason for that is that these producer strains often, they have many, many different biosynthetic gene clusters, and they produce something under condition 1 and something else under condition 2. The bioreporters can also help to detect polypharmacology. So here is an experimental compound where you see that several bioreporters are triggered. Or it can also help to detect synergism, as here when you see the sulfamethoxazole that is spotted closer and closer to the trimethoprim, and you see before you see any antibacterial activity for the sulfamethoxazole, you see the combination effect with the shape of the halo that is shifting. So we have these bioreporters not only with beta-galactosidase, we have them also with luciferase for different, and also with GFP for different applications. So if you have some, in terms of mode of action discovery now, if we shift gears in that direction and say, I have a new compound and I want to find out what the target area or the target may be, we basically always start with bioreporters in our lab now, but then we try to confirm with another equally easy assay. And for natural products at the beginning, you have only a tiny, tiny bit of compound. So often 1 milligram is like the world supply of compound that we get at the beginning, and we want something else. And here is something that I call microscopic phenotyping. It's basically the same. That is also called cytological profiling. But we didn't have all this elaborate machine learning afterwards. We just look at the phenotypes. This is always the same strain, Bacillus subtilis, treated with different kinds of antibiotics. And just by looking at the phenotypes, you can really see, is this a protein synthesis inhibitor, or is that a compound maybe inhibiting the membrane potential? It's a very nice confirmation. We are doing a lot of microscopy in order to further characterize the events that kill the cells, and we have lots of proteins labeled with the GFP for that. What you see here is the cell division inhibitor FDSZ. that is by a certain antibiotic or the mechanism of that it is degraded and it's no longer in its place. Here you see the penicillin binding protein 2. Very interestingly, if you would treat such a staphylococcus with penicillin, you would see a dislocation of the PBP from the septal side. So just also to tell you that lots about also very known and very old antibiotics we have never thought of and considered. Yes, that compounds then make the target go away from its normal position where it needs to do its job. And this is another opportunity, a time lapse of a molecule breaching the barrier of the cytoplasmic membrane. So this would be a movie, but I think I can't stop this. I can't start this here from this panel just with this. So I think it would be a time-lapse movie where you would see the very second of the death of the cell and see like how to a very certain point in time you see all the chromosomes that are labeled there. Can I start it? Can you start it for me? No. Oh, no, okay, sorry. Yes, so we would see that at a certain point in time, the chromosome just dislocates and then the cell ruptures and you could view it. And that shows you also the power of this high resolution microscopy that it's bringing today, that you can really resolve on the single cell level and view the moment of cell death. You can also do extract assays. Then you take off the barrier, and what you see here is a system that gives you information about a translation inhibitor versus a transcription inhibitor just by tweaking the system, by adding the T7 RNA polymerase from a virus. You see that rifampicin, which is a bona fide transcription inhibitor, is no longer acting on the right, while the negamycin, which is translation inhibitor, is working in both systems. And we have extract systems for other, like you can do it for peptidoglycan synthesis, for instance, if you are interested in removing the barrier. Yes, mode of action discovery platform, like decades ago, people were starting with radioactive incorporation of precursors. We have this in place, but we almost no longer need this because the bioreporters are giving us the information. Then, yes, I showed you the microscopy and examples of its power. Yes, there are available very nice and really cool full genome knockout libraries that you can make use of if you have some expectations of a certain target area and you want to narrow down. And it's not only the knockouts of the non-essential genes, you have also knockdowns of the essential genes. I showed you the extract assays. We have strain collections, clinical isolates, various, also very defined pairs where you can check on certain efflux pumps, for instance, and other particular pairs or that you want to investigate. A reference compound collection, Tuebingen, is home of about 1,000 natural products that have been by colleagues selected over the years. Yes, and there are, of course, then you come closer to the target and you have an idea of the target, and then you get down to the standard molecular biology in order to produce it recombinantly and to express it, and then to target interaction studies in order to do the biochemistry. All right, I get the signal that I'm done, and yes, I just would have wanted to add to the list another target, but I think many of you know this anyway, maybe from my name. The CLIP protease has been a target that has previously not been on this list, that has slipped the attention of the drug discovery programs in the companies because it's non-essential, but the antibiotic just activates and deregulates it so that also this non-essential protein can now be a very prime target. And if you are interested in that one, you can speak to me afterwards. I just slip this, my favorite target. And to tell you at the end, nature provides good starting points. And it has pre-optimized for many things, which makes working with natural products really fun. On the other hand, there has been a couple of things that nature had no reason and necessity to optimize against. And in order to reach the mountaintop, you have to really understand each target, each compound from the biology and from the chemistry in order to make it into a drug. But also, antibiotics are ideal tools for studying antibacterial cell biology and better understanding antibacterial targets. Thanks to my team, all the many great collaborators over the years, and thanks to our funding agency and 3 big collaborative structures that we have in place at Tuebingen. The German Center of Infection Research that Silke Alt has already talked about, but also funded by the German Research Foundation, 2 big structures that give us money to do fundamental investigations. One on the targets, which we call antibiotic cell map, and the other is the Cluster of Excellence, where we are looking at new natural products from the microbiome. Thank you.
So our next speaker is Carina Lundberg. She comes from the Lund University in Sweden and has performed research with animal models for the last 30 years. In the last 17 years, she's focused in on preclinical evaluation of antimicrobial interventions within mouse infection models at Statens Serum Institute in Copenhagen. Carina was the co-lead in the in vivo platform of the Enable IMI project and is now a part of the combined consortia within the AMR accelerator. Carina, thank you so much.
Thank you so much for inviting me to present our work here. So today I will talk to you about the work we're doing on standardizing a murine pneumonia model within the combined consortium, which is part of the IMI accelerator. So Combine is a consortium comprising of several different research groups, mostly in Europe, that are coordinated by Uppsala University. And we have a few scientific goals apart from the coordination functions. So, and one of those goals is to optimize and standardize an animal infection model to improve the translation of non-clinical efficacy to the clinical trial outcome. And our goal is to improve the understanding of animal infection model reproducibility and translation into the clinic. So the problem is that although animal models are widely used and they are a great tool for research, there is still some translational gaps that remains. And the methods used for performing these models will also impact the analysis of the results. And there is a lack of standardization that hinders interpretation and comparison of results between different labs. So our goal is to develop a standardized animal infection model protocol that will be benchmarked and standardized with relevant antimicrobial compounds across labs, and to establish an in vivo reference strain bank that can support this model. And it should also provide a framework for PK/PD analysis and mathematical modeling to, in the end, improve the understanding of preclinical to clinical translation. So we have started to validate the standard infection model, and the first goal was to select what model has the greatest needs. And we're now generating efficacy data in this model. And in parallel, we're also establishing a reference strain bank that will be made available to the AMR community. And in this strain bank, we will select candidates that generate robust results across different laboratories. So to select what model to standardize, we arranged a webinar in 2020 where Three of the partners in the combined consortium presented the work they are doing on various infection models that are used for evaluating new antimicrobials. And at the end of the webinar, we conducted a survey among the participants to select the model with the greatest need of standardization. And from this survey, it was obvious that most people requested a better standardization of the pneumonia model. And that's also relevant considering the global burden of pneumonia. And also that model is already now used a lot in preclinical research, both as a proof of concept model, but also to generate PK/PD data to estimate your pharmacodynamic target. So this is an overview of the process. We started out with a systematic review of the literature of murine models with relevant gram-negative bacteria. And we also had an open call for institutional protocols of pneumonia that other laboratories are using. And from this information, we identified the variables that would have the most impact of the results. And this was then discussed in an expert workshop, and the outcome would be the standard pneumonia protocol. So first, when we assessed the methods for acute pneumonia models with Klebsiella pneumonia, Pseudomonas aeruginosa, and Acetobacter baumannii, in the literature and in the institutional protocols that we received, we observed a substantial difference in the study methods used. So both in terms of animals used for the models, selection of strains for the inoculum, and how you perform the inoculation procedure, and also how you treat the mice and the endpoints. And if you want to know more about this literature search, we recently published a paper that you can see in the bottom of the slide. So if we look at these individuals, a few of them one by one, and this first one would be the choice of mouse strain then. We could see in the literature that both inbred mouse strains and outbred mouse strains are commonly used. And there is a long list of different strains. But if you look in the institutional protocol, there was slightly less variability there. There it was a majority of people using CD1 mice, outbred mice. Further, if you look at what sex is used, there is an overweight of studies using female mice. And also if you look at the age of the mice, that varies quite a lot, ranging from 4 to 10 weeks of age. And there we can see that using outbred mice, you tend to use maybe 6 to 7 weeks old, but using inbred mice slightly older, which is of course logical because they grow slower. And then the number of mice per group can also vary dramatically, ranging from 2 to 15. And there we could see that if you have bacteriology as an endpoint, you typically have 3 to 5 mice per group, whereas survival studies, you tend to have larger group sizes. So based on this, we proposed the standard of using female CD1 mice, 6 plus minus 1 week old, and you should use 5 to 6 mice per group. Next, we looked into the variable inoculation of the mice. So there we can see that both neutropenic mice and immunocompetent mice are used. Working with outbred mice, you have a more tendency of using neutropenia, whereas inbred mice strains preferably immunocompetent mice. We also looked into the method of inoculation. And there are several different methods used, but there is an overweight of using intranasal and intratracheal inoculation. And further, the volume of the inoculum varies, ranging from 10 to 70 microliters, 50 microliters being the most common. So for this, we propose to use as the standard immunosuppressed mice and inoculate intranasally with 50 microliters. Then we looked into the strains used for inoculating the mice. And there we could see that most people, they use their own clinical isolates or an isolate from a collaborator. Strain bank isolates are also used, but not very often. And there is also quite a high variability on how you prepare your inoculum. But commonly, people subculture them to a log phase. So for this, we have not suggested to standardize what strains to use. That will have to depend on the project and the specific needs. But we do advise that you use bacterials in an exponential growth phase. And you should use media as appropriate. Next, we looked into the variables concerning the treatment. And here we can see that the majority of studies describe that they started the treatment 2 hours post-inoculation. But there is also high variability here. And the baseline CFU is a very important variable, and that was only reported actually in 28% of the studies. But when we could find information, it was typically between 6 and 7 log 10 CFUs. The growth of the bacteria in the mice during the experiment procedure is also very relevant. And that was reported in the majority of the studies, but definitely not all. And the growth ranged from 1 to 3 logs, basically. And it's important that you do see some growth, indicating that you have a robust infection that you are treating. But setting the bar too high may then hinder selection of specific isolates that you want to use. So we propose as a standard to start treatment 2 hours post-inoculation, and that you should have a baseline CFU at 6 to 6.5 log, and that you should observe at least 1 log increase in bacterial loads during the study period. And then if we look at the endpoints of the studies, naturally most studies report bacterial loads in the lungs. But you can also report bacterial loads in other tissues and organs, such as blood, spleen, or liver. And also immune response and histopathology can be interesting to look at. And some studies have survival as the endpoint. So if you look at the duration of the study, then most studies run for 24 to 26 hours. But there are also various lengths. Many studies run much longer. So for this, we propose to have as a standard CFU per lung as the readout, and that you should end the study at 26 hours post-infection. So next, we arranged an expert workshop to discuss these standard variables that we suggested. And at the end, we had a survey to select the standard parameters. So this is the summary of the variables that we came up with for using the standard murine pneumonia model. And this has also recently been published in the paper in the bottom of the slide. And of course, there are additional consideration you need to take when designing your study. You may, for example, need to use both male and female mice in separate studies. And you may also need to adjust the number of mice per group based on the power analysis, depending on the virulence of the isolates you're using. And it's also encouraged to randomize the mice. And further, it's very important that you have a short time point between preparation of the inoculum and the actual inoculation to ensure a good viability of the inoculum. And we decided not to standardize the anesthesia procedure. However, it is important that you ensure that the anesthesia is deep enough so that the inoculum can settle in the lungs. And you should use an inoculum at least 20 microliters or higher. And if you need to run longer studies, it's advisable to include also the 26-hour time point in your study plan for comparison. And blinding the CFU counting is also advisable if possible. So now we are validating this standard protocol, and for this we are collecting strains for our repository. And we're receiving strains from partners, GNA Now and DSMZ, for example. So currently we have approximately 20 isolates of each of the 3 species. And these isolates are then used for the this infection model, and if the results meet the standard criteria, the isolates will be part of our repository. Strains in the repository will then be selected for further treatment studies in this model, and if we generate good data also with that, they will be part of the reference strain bank that will later be accessible to the AMR community. And here are some examples of the outcome of our validation studies. So currently, we have tested 11 Klebsiella pneumonia isolates in the standard protocol. And here I exemplify with 4 different strains. So if you look, for example, at the pink and yellow strain, the NCTC 13883, you can see that it starts at 6 log 10 CFUs and it increases with more than 1 log. And so does the yellow isolate. So that would be a suitable growth of the bacteria in this model. Whereas if you look at the blue isolate, you have a spontaneous resolution of the infection. So that's not a useful model, and this isolate will be discarded and not used any further. And the same procedure has been performed with the Pseudomonas aeruginosa. We have tested 12 isolates, and here's the example of 5 of them. So the top pink isolate, you have a nice growth. It works fine, and so does the yellow isolate. It works fine. But the blue and the purple, again, you have resolution of the infection, so that will not work in, that does not meet the criteria we need here. And if you want to know more about this validation process, please visit our poster. And lastly, I would like to reach out to you and ask if you would like to collaborate with Combine. So if you would like to share your expertise, share data, or share strains, please reach out to us. And if you want to conduct validation studies in your lab, please contact us. And with this, I'd like to acknowledge the collaborators in the Combine Work Package 5, GNA Now for kindly sharing strains with us, and the in vivo team at SSI for performing the studies. I'd also like to acknowledge IMI and the other funders of the program. Thank you.
Thank you, Carina. I hope that many researchers are reading your results and your papers, and that we have more use of validated animal models. So the last speaker today is James Sidaway. He's currently toxicologist at Apconics. He's a very experienced toxicologist and with many years, over 20 years of experience in the pharmaceutical industry and as an independent consultant. So we are looking forward to hearing more about the tox issues.
Okay, thank you, Ursula. Thank you for the invitation for the organisers. So yes, I work at Apconics. So Apconics is a preclinical, non-clinical safety company. It does a range of services ranging from ion channel screening for safety purposes all the way through to toxicology project management, and some of my colleagues do work on infection, microbial, antimicrobial products. I head up the target safety team. So this is, as you'll hear, this is primarily concerned with the safety of the primary target from the point of view of non-microbial therapeutics. So you may wonder why I'm up here talking, but before that I worked at AstraZeneca for many years, and I was involved in implementing a lot of hazard identification, screens and interpretation, looking at mechanisms of toxicity. And so I hope to bring you some of my perspective from that experience as well during this talk. So the outline of my talk is, I'm going to talk a little bit about where safety assessment fits in the drug discovery process, particularly concentrating on early safety assessment. I'm going to try and outline an overview of the antimicrobial mammalian targets, because obviously that is the main concern from a drug safety point of view. And by mammalian, I'm obviously talking about human targets, but also the non-clinical species that are used during toxicology studies. That's primarily rodents and a non-rodent mammalian species. I'm going to touch on the current safety de-risking approaches that have been adopted by pharma over the last 20 or so years, and then also talk about some of the newer approaches that are coming to the fore. And lastly, I'm going to say a very tiny bit about safety considerations of some of the new antimicrobial modalities which we see starting to come into use. And I guess underpinning all this, as I've already said, is my background is really a molecular toxicologist, so concerned with mechanisms of toxicity, applying molecular biology techniques and so on. So that's really my perspective for this talk. So if we think of safety assessment in the R&D process, we know that, as we've already heard, drug discovery, drug development is a very long process. From a safety point of view, we really think of dividing it into two halves. The first half, which is illustrated on the left-hand side, is what we call discovery toxicology. And here, there's a whole set of different assays and approaches that have been evolved over the past 20 or so years to identify the best possible molecule, give the program the best chance of success, identify hazards early, so fail early, so you can effectively design out safety risks while you still have the range of chemistry. The other side of toxicology is what we call regulatory toxicology, and this is really about designing the non-clinical package which supports a new chemical entity or biological entity going into clinical trials. So there's a whole set of regulatory requirements for that before a new entity can go into even healthy volunteer trials. And then as the product is developed, then there are lots of considerations around the nature of the patient population and other factors like, is it a combination therapy approach and so on. And a point I wanted to make is that a lot of the assays that have been developed over time have really occurred as a result of previous issues that have occurred. I mean, maybe that's an obvious thing to say. So at the moment, we're at the point where in pharmaceutical safety assessment, we have a set of high throughput assays, some in silico, some in vitro, that can be used to screen out hazards, safety hazards. But a lot of these have been developed over time based on rare adverse events that have maybe only occurred, or because they're rare, in the clinic or even in the post-marketing phase. And it's really through all the efforts of the scientific community, mixture of academia and industry, that's really started to identify mechanisms, and these have almost back-translated into new assays and approaches to allow further de-risking. So it's all a process. So important for my talk is just to understand where I'm coming from in terms of target-related toxicity and toxicity related to the modality. So of course the modality, as we know now, can vary a lot. It can be a small molecule, it could be an antibody, it could be a nucleic acid-based approach, it could be a cellular therapeutic. And the actual modality has a series of generic issues, so what I've called here modality-related issues. So for example, with small molecules, they can interact with secondary pharmacology or off-targets, chemical toxicity like reactive metabolites and so on. Genotoxicity can be an issue. There's physical chemical toxicity issues. Whereas a biologic, so that's the other three modalities shown here, because they're biological in nature, they tend to have more immune-related side effects, so particularly immunogenicity, so that's a response of the patient or the non-clinical species getting an excessive immune response. And then they have other more focused issues as well, which I won't go into today. On the other side, we have what relates to the modulation of the target, so what we call on-target toxicity. So at its simplest, this could be exaggerated pharmacology of the target. So you inhibit a target, you inhibit its function, that results in some unintended consequence. And then there's various variations of that. Inhibition of the target might exacerbate a disease. It might act on a different tissue to where where the intended therapeutic mode of action is, or there may even be an interaction with a closely related target. So if we just turn to conventional antibiotics, I've done a list here of issues based on a literature survey of some key reviews about the sort of issues that have been historically encountered with these various classes of antibiotics. And again, as I'm sure the audience appreciates, safety can have serious consequences. So in the 90s, there were several new drugs that were withdrawn, either due to cardiac effects, hepatotoxicity, or other immune-related effects. And the point was these were discovered in the clinic or once the drug had reached market. More latterly, there are effects that have stopped the development of certain candidate drugs, and then there are just toxicities that limit the use of current antibiotics in terms of clinical use. On the other side, here's an analysis of some more recent examples in the pie chart, and it's interesting to see that there are really many of the same safety issues still occurring, and obviously there are other non-safety reasons as well that have stopped the development of these candidate drugs. So the current safety de-risking approaches that are used by pharma, I'm going to touch on some actual case examples to go into a bit more detail, but in the discovery phase, there's a battery of, if I go back a step, first of all, I'm going to talk about the target safety assessment, and I'll try and explain why I'm talking about target safety assessment to this audience in a moment. Then I'm going to talk about the in silico in vitro safety liability screens at quite a high level, but the type of screens that are widely adopted by pharmaceutical industry are screening for mutagenicity and high-throughput Ames tests, for example, screening against the Hurg ion channel, which is a cardiac ion channel. If you block that, you can get an arrhythmia called QT prolongation and ultimately result in sudden death. There's then a series of other assays that have been developed that monitor cytotoxicity, phenotypic outcomes, looking at mitochondrial toxicity, which I'll also come to in a moment. So an important thing to say is, although I'm just going to keep talking about toxicology and safety, toxicology is just part of drug design. So clearly, the drug metabolism, pharmacokinetics is very important. The therapeutic response in terms of the microbiology is obviously critical, and the chemistry itself. So one of the previous speakers was talking about the relationship between chemistry and the target. So all these four properties are important to get the balance right for a successful drug. So just to go to the first part of that pipeline, a target safety assessment. So this is something that's used quite routinely in the pharmaceutical industry. And basically, it considers the biology to do with a novel target that allows you to try and work out what the possible target-related toxicities might be. So for a novel drug target, so this would be a human target, you'd be looking at genetics, you'd be looking at the expression of the target, you'd be looking at pathways, biological function, doing a wide literature survey, bioinformatics review, to really try and infer what are the possible risks. But obviously this approach could be applied to an off-target as well, and I guess this is the point of where I'm coming from antimicrobials, as I'll say in the next slide or two. I think this understanding the off-targets is clearly very important too. So, if we think about antimicrobial drug modalities and the mode of actions and how they might interact with mammalian targets, I guess there's some obvious points to make. So conventional antibiotics, you don't really have to worry about the primary human. There isn't a primary human target, obviously. But we know off-targets, mammalian off-targets, are a possibility. And if we just go down where we would have to be concerned about the primary target, maybe if you're using repurposed drugs that have come from, that have been developed for other indications, other diseases, possibly if you're looking at trying to modulate host factors through immunomodulation, you'd be concerned about the primary target and I think you'd want to have one of those target safety assessments. For all the other modalities, off-targets are obviously a concern, apart from possibly some of the genetic and phage therapies. I guess that's not really well understood at the moment. And again, that may occur, finding that information may occur later in the drug discovery development process, but again, commissioning a target safety assessment to try and understand biological consequences would be very useful. So if we think about mammalian off-targets of antimicrobials, what are we really talking about here? And I think I'm probably overlapping a little bit with one of the earlier speakers. The way I thought about it is we're talking about several different types. So we've got evolutionarily conserved targets that are related to the microbial target. So that might be based on a conserved domain of function, and mitochondrial targets are an obvious example. There's targets that aren't obviously related to the microbial target. So GABA receptors, the hERG ion channel, they're not obviously related, but they are known to be off targets of some antibiotics. There are other mechanisms that aren't like classical pharmacology. So you can get chelation of key ions, which might affect certain target proteins. And then other antibiotics have been known to cause exon skipping, so have genetic effects. Then there are other considerations. It seems to be that, if you like, poly-safety pharmacology is an issue. You often find multiple targets are interacted with by the antimicrobial. It's probably a common finding. And then there are other factors that don't necessarily relate to targets themselves. We know that the host microflora can obviously change in response to the antimicrobial treatment. And again, this can impact safety assessment and adverse events. We need to relate the high concentrations of the interactions actually with the off-targets to the actual exposure of antibiotics when they're in the non-clinical model or in the clinic. And of course, many of these off-target interactions from a mammalian point of view may actually be benign. They may just be a binding. They don't actually cause a modulation, or they may even be therapeutically beneficial. So what tools are available to look at mammalian off-target assessments? This is a bit of a montage of different approaches. So well-established are in silico screens, QSAR models, and so on, but also other modeling approaches. And if you like, this is really divided into what would be the established safety targets and novel safety targets. And I've just marked them up as to where you'd expect to apply them routinely, or maybe you'd apply them on a case-by-case example, maybe as a kind of follow-up strategy to identify a mechanism. A mainstay of the pharmaceutical safety screening paradigm is conventional secondary pharmacology screening. So there's commercially available panels of targets, safety panels, 40 to 80 targets with reasonably well-established safety endpoints. So if you hit that target and you modulate it in vivo, you may actually see an adverse event. So those probably should be routinely screened for. But then there are larger superfamily screens that are available that have just evolved through drug discovery, like kinases, GPCRs. There may be times when maybe if you get a hit from the smaller panel, you may want to follow up and look at a more diverse panel. And then there are open-ended, label-free molecular screening approaches. And again, this is far from an exhaustive list, but there are examples of how the thermal shift assay, or CETSA, has been applied to discover off-targets of some drugs that have been linked to safety findings. And then there's also chemical proteomics, where the small molecule is modified and used to capture its putative target. So again, these are approaches that you may employ on a case-by-case basis. And then just to not forget some of the other modalities. So for biologics, so I know that there's some antibodies, monoclonal antibodies that are under development or have been approved for as part of this whole area. There's a well-established platform where if you fractionally label the biologic and you apply it to a protein or cell array that expresses thousands of human targets, you can get an idea for off-target binding, because that can occur with antibodies. Targeted protein degraders or bacterial PROTACs, which I'm going to mention a bit towards the end, because the TPD area is really growing in oncology and in some other therapeutic areas. Farmers developing, or academia, approaches to look for off-target effects of this class of drugs, initially by Western blotting of sentinel proteins, but more latterly by mass spectrometry proteomics. And then genetic therapeutics, you could apply mRNA sequencing, look at monitor the changes in exons and so on. So mitochondrial toxicity keeps cropping up. It's clearly probably the most common mechanism. I don't think I need to go through the figure on the left, but so if we go for the bacteria to the mitochondria, which I guess is a bacteria of some sort, or was once, you could see how various antibiotics target pathways or off targets within mitochondria, resulting in mitochondrial disruption, dysfunction, and some of the potential targets that have been described, which are very similar to the therapeutic targets in bacteria, are listed below. Important thing to say about mitochondrial toxicity assessment. In pharma, there are a range of HTS assays in cell lines, but I guess what you call is that they're a compromise because they're high throughput in nature. So they're often based around fluorescent imaging. The cell lines are often cultured in galactose to make them more sensitive, because you're making them less glycolytic, more dependent on the mitochondria. And not all mitochondrial mechanisms are detected in this approach, but I guess it picks up a lot of mitochondrial toxicities. Also, the value of using a primary or 3D culture, so maybe human or from a non-clinical species, using the seahorse type platform. Although this is lower throughput, this gives more organ-specific sensitivity. So I've got some examples about de-risking chemistry. So this is moving back more to the more established techniques and approaches. So in the example on the right shows, I think that's from 2001, shows a quinolone SAR relationship, and with each of those residues you could see particular interactions that have been defined. So that just shows the level of understanding. So you can modify GABA binding, for example. You can modify genetic toxicity and phototoxicity. So a lot of this SAR, at least for particular compound series, is well established. In terms of in vitro screens, as I've already mentioned, you may use screening against cardiac ion channels, particularly the hERG ion channel, secondary pharmacology, genetic toxicology, mitochondrial toxicity. We then head towards the animal studies. So in the first instance, this could be efficacy studies in which you could add on some toxicity endpoints, then proceeds to the dose range finding toxicity studies and investigated toxicity studies, where you'll be pushing to higher doses and maybe doing a deeper analysis. And again, it comes back to what concentration should be attested for antimicrobials. I guess they tend to be quite high concentrations, exposures, doses. So this has to be done at a relevant dose in these models. In terms of different examples, I've got 3 case studies to talk through now about how particular safety liabilities have been de-risked during the drug discovery process. So in this example, this pharmacophore is associated with cardiovascular liabilities. And what you're seeing here is data from an animal cardiovascular study looking at heart function. And basically, this is an in vivo marker for hERG inhibition. So by modifying the chemistry, we were able to, or the authors were able to de-risk this toxicity. In this example, this compound has promiscuous binding to a whole range of human secondary pharmacology targets. And you can see on the plots on the top left-hand corner, the free exposure and the selectivity against each target, and how this was encountered within the toxicity studies. That's what those different lines illustrate, that exposure. And by improving the in vitro selectivity, a follow-up compound had an improved in vivo safety margin. And lastly, in this study, this is looking at genotoxicity, and two stereoisomers were tested, and essentially the authors were able to mitigate the mutagenicity while still maintaining the microbiological properties. So moving on away from the established models, where are we heading? I think over the past 10 years or so, there's been adoption in pharma of more advanced in vitro models. So if we think we started off with the cell line, we're using primary culture, which might be very physiological relevant, but the cells would generally have poor long-term viability. More recently, we've had access to stem cell derived, so induced pluripotent stem cell models from which you can derive cardiomyocytes, neuronal cells, other cell types. And this has allowed us to access cell types that are more viable long-term than primary cells, although one issue is they still often have an immature phenotype, although many seem to be adequate for pharmaceutical testing. The next level is going to 3D, where you can either use the primary cells or the stem cell-derived cells, where you find the organization at a tissue level gives even greater physiological response, and it's very good for long-term tissue culture. So all this is about making more relevant in vitro models that are more organotypic for toxicity testing. And how are these applied in terms of drug safety assessment? Well, essentially, you can apply them at different stages. So if you're looking at a primary target issue, you might take your organotypic model very early on in the drug discovery process. You may try and knock out the target in that model to assess a risk, whether that's pharmacologically or genetically. Later on, these models have real value in terms of mechanism ID, testing hypotheses, and doing what we call in vitro translation. So a common feature in toxicology is you'll get a species-specific toxicity, and the question is, will it translate to man? And there may be biological reasons why that doesn't happen. So if you can have in vitro models from the key species, including human, you can do an in vitro translation. So that's really where the field's heading. Final point in this part of the talk is talking about mechanistic modeling. So this probably answers the question about how do we go from hazard looking to risk? How do we take in vitro data and essentially make predictions about what could happen in the non-clinical animal studies or in the clinic and in particular patient population? So this whole area of quantitative systems pharmacology and toxicology is a real growing area. There's models being developed for various types of organ toxicity. This is one of the earliest examples to come out, DILI-SIM. So this is a model of drug-induced liver injury, DILI, that was developed by the Hamner Institute and a pharmaceutical company consortium. And in the example shown here, liver enzyme elevations were seen in the clinic, so in early clinical trials with this novel antibiotic. In vitro assays were performed, including mitochondrial assays and other mechanistic assays of drug-induced liver injury. This was all put into this mechanistic model, and from that, predictions could be made. So first of all, predicting what was the most likely mechanism, which turned out to be mitochondrial, and what would be an optimal safety dosing regime to maintain an adequate safety margin from the drug-induced liver injury. So as I say, this is a real growing area in how early data or data from more advanced in vitro models can be taken, modelled, and to do an improved risk assessment. Last two slides to talk about. I said I'd talk about some of the newer modalities. So I'm just trying to put this in context with how some of these modalities have operated in other therapeutic areas. So monoclonal antibodies, there are 3 endotoxin targeting monoclonal antibodies that are approved. In wider pharma therapeutic space, there are numerous monoclonal antibodies that have been developed and approved. Common safety issues for those antibodies include immunogenicity, which I mentioned earlier, and some binding to off-targets, which can cause toxicity. So if we look at the 3 drugs that have been approved, the clinical studies suggest that the safety profile was well tolerated and benign, no obvious safety concerns, so no significant immunogenicity or off-target binding was reported. And that may be a reflection of, I guess, the understanding of how to design these molecules. So maybe issues like immunogenicity have been actually taken away, but I guess that is encouraging. And this slide just talks more about these targeted protein degraders. So in drug discovery, there's this principle of basically targeting a protein of interest and then pulling in the cellular degradation protein. So in mammalian systems, drug discovery is focused on the E3 ubiquitin ligases, and there's a particular subclass of targeted protein degraders called PROTACs that have this property, and at the moment several of them are in phase 2 clinical trials in oncology. The issue is, though, that the ligase they target is called cereblon, and this is heavily implicated in thalidomide teratogenicity. Now, for obviously an oncology indication, if that risk probably wouldn't manifest itself because of the patient population, but for other indications, this could be a serious issue. So I've noticed that the same approach has been applied is basically in the early stages for antibiotics as a new approach, targeting this CLP-CP protease. So it's not an E3 ubiquitin ligase. You wouldn't expect it to be in bacteria. So this is obviously an exciting new technology for next generation antibiotics. So because it's not cereblon-based, that risk of teratogenicity, at least in theory, is removed. So really the other hazard that's associated with PROTACs, like any small molecules, and these are obviously large and bifunctional, is interaction with other off-targets, but I guess that's no different from the other small molecules. So just to summarise, I've told you about how current safety de-risking approaches can be used to mitigate against common antimicrobial toxicities. I've also talked about the importance of understanding the off-target toxicities, the mammalian off-target toxicities, how this can help the process. I've touched on the emerging safety de-risking approaches, which hopefully with time to come will help to improve resolution of more complex safety issues. And I've also briefly talked about the safety considerations for the new microbial modalities. And this is obviously a developing area. It's probably too early to have a firm opinion on whether there are any specific safety issues, but based on what we've seen so far, it all looks quite encouraging. So just like to acknowledge my colleagues, Claire Sadler and Professor Ruth Roberts, And thank you for your attention.
Thank you very much. I would like to invite the speakers to come up for the discussion. So, I hope you found it interesting and have a lot of questions to discuss. Maybe I should start with a question addressing Heike. My feeling is that after this, that we have waves in drug discovery. So first we started with natural products, and then we had the target-based approaches that were not successful. Then a little bit, again, discussions of maybe going back to natural products. And now I think that we are, again, back to target-based approaches. I don't know if that's just my feeling, or what do you think? Maybe we have also new, very new technical means of doing target-based approaches compared to 30 years ago.
Yes, I think there is no such paradigm again, like that is promoted as the one particular path to follow. I think nowadays everything is allowed. And what you mentioned is like there have been this like natural product like or more antibiotic like libraries, focused approaches. Now people have better means of studying just binding to targets of interest. Fragment-based approaches have been followed all along. And now with increasing knowledge into how molecules can be made cell penetrable, I think there are other opportunities too. But I think also on the natural product end there is continuous progress, and probably I'm more involved in this particular field. So there's lots of genome mining approaches, metagenome approaches, and there are now the first molecules being pulled out of this endeavor that show promise. And the natural product people looking at the gene cluster still tell us that there is like just about 1% of the gene clusters that we have accessed so far. So I still hope that there is opportunity too in that area.
That would be good news, that we don't have really these pronounced waves of one specific way of doing drug discovery, but spread it.
Yes, I get the impression that there is much more. I mean, who is to prohibit?
No, it's not about prohibiting, but there are some
trends that's set like that's the stage.
I wanted to say fashions, but it's more trends. Yes.
Yes. Yes.
More questions? Yeah, please.
Sven Hobby from Zurich, Switzerland. Carina, thank you very much. Very nice talk. I think it makes a lot of sense standardizing the models. But the second part that you kind of mentioned was the predictability with regards to clinical outcome. And you haven't spent much time talking on this. Is this something you're still going to engage with after you standardized to see, like, can you actually tweak some of the protocols with regards to what we see ongoing in human or in clinical patients? Thank you.
Yes, so that's activities that are ongoing within the combined consortium. So we have an open call to get clinical data so that we can compare the clinical data to the data we can generate in the models. It's proven difficult to receive this kind of data, but we're still working on that.
Sorry, just because it's fitting this direction. How did the very first animal models compare the first models that were used for the PKPD studies 20, 30 years ago? So did you include them, or did you just take the recent?
Yeah, so one inclusion criteria was actually studies being performed in the last 10 years.
Okay.
Yeah.
Please.
Hi, thank you all for your interesting talks today. I'd like to ask, probably for all the speakers today, a general question about in vivo models of antimicrobial resistance. Firstly, in vitro, how that could be investigated further. I know that there's a lot of work going on at the minute, but also in vivo, again, any possibility of standardizing models. There are models out there, but again, how standardized are they? And finally, touching on the safety aspects of those as well.
Yeah, so there are no standardized models for investigating the in vivo generation of resistance, unfortunately. So are these typical in vitro models with serial dilutions, for example, that you run for 14 passages or so. You can try to do similar studies in vivo, but it's not validated how predictive those types of studies will be.
Do you get this high inoculum in animals that you would need?
That is exactly one of the problems, that in these small mice it's difficult to have high enough bacterial loads to observe development of resistance at a low frequency.
So without standardization, is there a need to have such data for pharmaceutical industry regulatory packages, for example?
Yeah, I mean, there they describe that they want to see data, for example, in the thigh or pneumonia model, but they do not specify exactly how that model should be performed, and they do not require specifically a standardized model. But we're doing this standardization process to help people get started with their in vivo efficacy studies. And it may not be able to cover the whole need, but at least it could be a good starting point.
Do you see any safety implications from investigating these type of studies in vivo?
I think it would be useful maybe to predict possible issues, to maybe include additional biomarkers, histopathology, if the efficacy studies are being run. Or for the regulatory submissions, it's normal, healthy animals that are used. But the principle of doing some form of safety assessment and efficacy studies is something that's been done before. But I don't really understand the resistance issue enough to be able to comment from a safety point of view.
Thank you for your questions, actually, because it reminded me of something that I was thinking about. So with the standardization, do you have a length of time that you're going to standardize the model to run to? Because with a survival model, for example, we let our models run anywhere from 3 days to 4 days, but sometimes we let them run for another week. And then you'll see animals maybe pass later. And at that time, when you have more time, then you have more time for resistance to develop. So that kind of gets maybe at the question that was there. So maybe the numbers aren't necessarily high enough, CFU in the lung, for example, but if you give it more time, resistance can be there. So if you're thinking about standardizing, that might be one issue. How long will it be?
Yeah, so this endeavor of standardization was primarily focused on the 26-hour acute infection models that is used for setting the pharmacodynamic targets. With having the CFU read out at 26 hours.
So you're just measuring CFU, you're not doing survival.
Exactly. So then if you want to measure survival as an endpoint, then I totally agree with you that you have to run the studies for much longer.
Okay.
And that would be a separate standardization process that
separate from
take on after this.
And then the other thing, I just have a quick comment, because your talk, I was really interested in how you had the BAL drug that had the liver signal. One of the things I was thinking about is all the failures over the years in this business. It'd be really nice to put them back into animal models in different things and see if you can catch the failure earlier and use that to help with standardization and some other things. Because if we know those failures earlier, we can then apply it to things later. So.
Yeah, that's a really good point, and I guess that's in some way the way things have evolved. We learn from those failures. I mean, there's certainly been a lot of research in the toxicology community. There's been IMI projects and so on looking at drug-induced liver injury, but it's very complicated. The idiosyncratic toxicity, it's down to genetic factors, environmental factors, and I mean, there are some that I can think of that we still don't understand the mechanism, despite throwing the whole kitchen sink, as it were, at them.
I was waiting for that now, please. Yeah, tell us your story. Oh, sorry, yes, you are first.
Hello, Mikkel Johansen from Aarhus, Denmark. So, yeah, thank you for some very interesting talks. Carina, I was thinking, this way of standardizing animal models or developing animal models, is this something that you think could be applied to other animal models and maybe other animal species?
Yes, of course. Any animal model that you want to use should be thoroughly validated and in a way standardized. And here we're trying to standardize across the community because we have seen and we know that many different labs use these types of models. So then it may be useful to have a standard to refer to. You may not always be able to use exactly that standard, but at least you can refer to it and say that I had this modification. And I would say the same holds true for any model that you want to run. If there are several labs running similar models, it may be wise to communicate and try to harmonize the procedures if you want to be able to compare your data and if other labs want to reproduce your data.
Thank you. And one more question. So one of the problems that we have found with animal models or in vivo models is that for rodent models, the combination of the rodent's very strong immune system with antibiotics is very hard to translate to the clinic. What do you think about that? Do you think with your model that this is something that you could overcome?
What did you say, the immune response? Or?
The immune response of the very strong immune response of rodents combined with antibiotics. So what we see is that we can cure an infection in rodents that we would not be able to cure in humans. So how do you think that we can overcome this?
Yeah, that's, I would say, part of the translation problem, that sometimes mouse models are good in predicting what's going to happen in humans and sometimes they're not. Mm.
Thank you.
Maybe one comment to this, Carina. Isn't it that you have always these comparator antibiotics that you use as benchmarks, and will they help you in that regard? Like to assess how potent a compound needs to be in order to cure the infection. You always, I think, use benchmark antibiotics in your models.
Yes, so exactly. So we use control antibiotics where you expect a certain effect, yeah. Mm-hmm.
Ryan?
Yeah, there it goes. Ryan Source from Revigenics. First, a comment, just thank you for standardizing. As a sponsor, I'll do whatever you all tell me. I just want to do it once. And that really helps just to have clarity around how things should be done. The one thought I had to share, and it's not a practical solution for everybody, but it's the one time I encountered a way to look at resistance, is really in the biodefense models. So when we were running those models frequently in rodents, but especially primates, there you're talking about burdens of 10 to the 10 per gram of tissue, and many, many grams of tissue in a primate before you start treatment. And treatment needs to be effective for at least 5 to 7 days. We even looked at duration, and you'll fail if you stop treating early. So if resistance emerged in the first 5 days, you would fail. Obviously, the studies are $1 or $2 million. It's not practical, but in a world where if you're developing with that in your program, that's the one place where I felt really good, especially with a novel mechanism drug where we didn't have clinical information to know the class could stand up over decades. That was the one place I felt like we were getting a look at in vivo resistance suppression.
Am I on? There we go. Don Shepard from McGill University in Montreal. So the NIH actually sponsored in the fungal field the development of standardized models of fungal pneumonia, which I was involved in. And we learned some very painful lessons during that. And the first was selecting our standardized model, because I didn't quite follow the criteria you used to select it. If we did the vote by what you do method and came up with a consensus protocol, which is what it sounded like you did, it was actually horrible. And it was horrible for intra and inter-laboratory variability because what people were doing out of habit didn't actually turn out to be what was very reproducible. So I guess I'm questioning how you arrived at your standard protocol. And I saw you're breaking down by strains, which is clearly obviously necessary, particularly in the Pseudomonas space. But are you also looking at some of those other variables? For example, intranasal turned out to be a disaster in our field because my intranasal, your intranasal, and my grad student's intranasal gave us 2 log difference in inocula with the same mouse and the same tube and the same pipette. We actually had to move to intratracheal through the mouth to get any form of reproducible inoculum. So I guess the sort of 2-part question is, how did you get where you got, and are you going to try and break it down? Because I think the inter and intra-lab variability issue is what's really critical if you want to move the field forward and, as you said, start to compare data sets.
Yeah, I agree with you. That's the critical differences. But we discussed these parameters in an expert workshop, and we reached a consensus, for example, on the inoculation procedure. But as I also mentioned, we did not standardize how to anesthetize the mice. So, for example, now when we are running our validation studies, we are anesthetizing the mice. They're sleeping for 4 hours, and we get a robust inoculation. The other labs that are going to reproduce the data, they also use intranasal, but they typically use isoflurane, so they sleep for a very short time period. They also get robust inoculation and deposit of bacteria in the lung with their method, with the strains they're using. So now it remains to be shown that we can get the same results with the same strains. And optimally, we would like to also try to work with these parameters and maybe try various methods. For example, if we try a different anesthesia method, does it work also in our laboratory? So we have just started. We'll see how it works out.
Over to the left.
Shampa Das from the University of Liverpool. So I just want to come back to the question about how we're going to look at emergence of resistance. And I think there's some important information said that the rodent model isn't well set up for this. Maybe the primate model is a little bit too difficult to do. One of the things that we're looking at the moment is whether we can use a rabbit model. So, longer model, we can go in at higher inoculum. And that's still work in progress. But the thing I just wanted to raise, especially for people new to this field, is that we do use the hollow fiber infection model. So it's an in vitro model and certainly doesn't have the characteristics of in vivo. But if you use it together with the in vivo model, we can go up to those very high inoculums and we can test the system out for a lot longer. So, it's not an ideal solution at the moment that solves all problems, but certainly we are able to look at emergence of resistance with these models. And, there's quite a few people doing that at the moment.
Yes, thank you for this comment. It brings me to one point, that there is maybe not only the in vitro test tube and then the mouse, because there will be a gradual range of different models serving in between, also more humanized cell culture-based models, organoid models, if you consider even microbiome occupancy later that you can put in place in such models. So I think in the years to come there will be more range of different models where you can ask different kinds of questions.
Hi, thank you all for great lectures. Thomas Walsh, USA. In addressing the question on establishing animal model systems and multi-drug resistant bacteria, if you look to clinical trials, they're extremely difficult to accomplish. And many studies have gone forward and not achieved regulatory approval for small numbers. One way to augment the credibility and efficacy of a given antimicrobial compound is our robust animal systems, but not the animal model, but several complementary model systems that then can collectively be able to support a smaller limited trial, but with with significant, robust preclinical data that also de-risks the potential for even going forward with a clinical trial. But how to do that? And certainly the standardized mouse model, variations on which are tried and true and tested, but with considerable limitations, not the least of which is we start antimicrobial therapy on our patients 24, 48 hours likely after the infection was established. And when you see pulmonary infiltrates, which already indicates organism-mediated pulmonary injury, which is certainly not at 2 hours, but certainly the workhorse model, murine model that Carina, you and your colleagues have very nicely developed, is a start. But what can be helpful are some of the other strategies of other complementary systems. Beyond mice, we've taken an approach toward large animal or intermediate animal systems, and the FDA had a large seminar underscoring the limitations of all models, of course, including murine models, and advocated for the intermediate models. We've gone to rabbit model systems and have developed over the course of time multi-drug resistant Pseudomonas, Klebsiella, Acinetobacter, and Stenotrophomonas, addressing some of these problems, one of which is inoculum. We use 10 to the 5th to 10 to the 6th inoculum. We can go up higher, but of course have a more rapid collapse, with the strategy of colonizing the tracheobronchial tree. And you colonize the tracheobronchial tree, come in with immunosuppression, very aggressive immunosuppression to address this very important question of role of host response. We aggregate the host response, there is none. And if you look at our poor patients in ICU, they often have very minimal host response. And as immunosuppression ensues, the colonization then evolves from colonization over the course of 12, 24, 48 hours into a progressive pneumonia that we're able to document radiographically as well as by serial histology. And we can carry out with very aggressive supportive care the animals out to 10 to 12 days by allowing this question of can we detect the emergence of resistance? It also helps, Dr. Sidaway, to help us evaluate toxicity as well during that period of time. So if we envision how we might be able to move forward with limited data, small populations with multi-drug resistant bacteria, we can see a complementarity of several animal model systems that collectively could support that clinical trial.
The workshop was an excellent workshop I attended, the animal model workshop. And so a lot of really amazing science was presented, pigs, rabbits, all kinds of different ideas, all really, really good models. At the end of it all, I said, if we did every model we talked about today, name one thing I don't have to do in the clinic because of that. And there was nothing. And so it doesn't mean we don't use them, but I think we tested that with the regulators. If we do all of these other things, what can we do differently in the clinic? And the answer at the time was nothing. And so at this point, we're still treating the models, at least from my perspective, as I want to reduce the chance I fail, but I'm ultimately going to have to do everything the same in the clinic. And that may be revisited again as things evolve, but that was the ultimate conclusion of the workshop. Yes, you can do all these other things, but you can't do anything less than you were doing already.
If I could address that, it seems that in anti-infective therapy, we are extremely limited and often forced in rare diseases. And if you consider multi-drug resistant bacteria as dangerous as they are, numerically they would still fulfill what we call uncommon or rare diseases by sheer demographics. If you look at oncology, certainly at FDA, my colleagues and I have gone through all of the approvals of agents for rare cancers. And these are rare cancers, not small cell lung cancer, but uncommon cancers, rare cancers, that were not based upon large, multi-center, randomized controlled clinical trials, but based upon much wiser designs, open label, that were then compared, the efficacy of which was comparing the registries, historical controls, expert opinion, but there are well over 25 rare antineoplastics developed for rare cancers over the last 2 decades, and just for cholangiocarcinoma, an absolutely deadly cancer, FDA just approved on the basis, just last week, 109, based on, I believe, is 103 cases with a 43% response rate, open label, knowing that that case, a case of cholangiocarcinoma, is almost universally deadly. So why is it that we may not propose to our regulatory colleagues to look to oncology, rare diseases, and I would also mention, but won't go into it, inborn errors of metabolism, where we also see a smaller set of compounds that have been developed without the large randomized trials, and perhaps open up a refreshing dialogue to say, maybe we could look at other models that have been developed in a structured regulatory basis, still fulfilling the codification, the regulatory basis of having a well-controlled clinical trial. But the question mark then comes on control, that we may not necessarily have to be constrained into the classic randomized double-blind trial.
Yeah, and I appreciate the comment. I mean, Acagion was the poster child for attempting the type of trial you were just talking about, I mean, effectively. It took us 3 years and $1 million per patient to attempt to show mortality superiority. And, you know, I'm panning for Dr. Rex to back me up. This is just an opinion, but I think the one reason we don't do that in antibiotics is we have another path. I think for those other drugs, there wasn't another path. For drugs that use LPAD, there was only one path. We attempted to use LPAD for Zemdry, and there was another path, so we didn't get to use it. And there is a path. It's just, unfortunately, nobody likes it, but it's a well-controlled, randomized, non-inferiority HAP/VAP or UTI study, and then use all of the non-clinical evidence to demonstrate superiority. I think the existence of that path makes us not able to do what oncology can do, my opinion, and I'd love it to change. And everything you're saying scientifically, I 100% agree, but I've sort of fooled myself into thinking we could do it. We tried for years and years, we failed, and ultimately we came up with a path that I think works, but we just have to get clinicians to understand the trial itself is not the place we show our superiority, and that's been tough, and it's been tough for pricing in Europe. It's why the drug I worked on withdrew its European application after it was ready to be approved, and in the other hands for a different company.
So thank you very much to all of you for your contributions, and that you would stay with us for the whole afternoon. And I also would like to thank to the organizations which contributed, that it's GARD-B, Repair, Impact Fund, CARB-X, GPI, AMR, Action Fund, and Orcam Trust. So thank you very much for staying with us, and I'm wishing you a really interesting next few days, a good conference, and we are all meeting outside this hall now for this networking reception. See you all there, and we can continue our discussions there.